Hi, everybody. So this is the alternate version of our evolution and social issues unit about antibiotic resistance and explaining the emergence of antibiotic resistance as a really serious human health crisis through the lens of evolution. So thinking about how the antibiotic resistance alleles initially emerged. So why did those alleles occur? And then ultimately, why have they become an increasingly serious problem as they become more and more frequent in various strains of bacteria? So this is the alternate version of this unit. The documentary that we watch, Hunting the Nightmare Bacteria from PBS Frontline, is freely available online if you want to watch it on your own. But if you're sensitive to medical imagery or have some reason that watching medical imagery is particularly challenging for you, that's okay. So for that, what I've done is create this alternate version where what we're going to do is give you all of the information that you need from the more challenging portions of the documentary so that you don't miss out, but can also do so in a way that's respectful of any potential difficulties that you might have with those images. So let's talk about the way that this is going to work. For the front half of the documentary, that's where some of the more challenging images might be. So the way that that's going to work is that I'm going to essentially tell you the story that's depicted in that documentary and give you all of the more in-depth scientific analysis of exactly what's going on in those scenes. The second half of the documentary isn't so much about the actual medications and treatment plans itself, but instead is more about why we have this problem in the first place of not having new antibiotics to treat these really challenging to manage antibiotic resistant strains of bacteria. So we're going to go ahead and jump in, and you'll get everything that you need from this unit without watching those challenging scenes. So the key focus of this is the idea that antibiotic resistance is becoming an increasingly serious global human health threat. To give you an idea of how serious it is, I've got this quote from the Assistant Director General for Health and Security at the World Health Organization. And they say, without urgent coordinated action by many stakeholders, the world is headed for a post-antibiotic era in which common infections and minor injuries, which have been treatable for decades, can once again kill. So that's a really serious statement because for all of us who are thinking about this nowadays, we've lived in a world where antibiotics have been readily available and able to cure our illnesses. So if you get a little cut on your arm, no biggie, rub some Neosporin on it, you're all better. But if we rewind the clock less than 100 years, so back to right around World War II, antibiotics didn't exist. So people who got minor injuries could have those injuries get serious infections and then potentially die from it. So what we're talking about now is that we're losing those tools we had to fight bacterial infections. So we're going to think about why that's happening, and then ultimately our thoughts about the ethics surrounding solving this process through development of new pharmaceutical inventions. Okay, so the first thing that we're going to talk about are two case studies to understand how antibiotic resistance has affected people in distinct ways. So what you're going to do as you're listening to my description of the stories involved in that documentary is go through and answer these study questions as I'm describing what you would have seen in the documentary. It's going to go straight through in order, and then we're going to kind of do a recap where we talk about the science that's described in those stories. So the first story in the documentary involves a little girl named Addie. Addie was a perfectly healthy 11-year-old girl who had no real underlying medical conditions. But then Addie got a little injury, and she started feeling really, really sick and got really sick really quickly. And eventually, once she was fevered and achy and seemed pretty severely ill, her mom took her to the hospital. And eventually at the hospital, they diagnosed Addie with community-associated MRSA. So let's break that down. MRSA, MRSA, stands for methicillin-resistant Staphylococcus aureus. Methicillin is a group of antibiotics, so they're the methicillin class antibiotic. Methicillin resistant means that those bacteria can no longer be treated with that methicillin antibiotic. Staphylococcus aureus is just the type of bacteria. So if you've ever heard of a staph infection, that's what we're talking about, Staphylococcus aureus. So the doctors figured out that Addie had gotten community-associated MRSA. Now, the community-associated bit is particularly important because when we think about antibiotic-resistant strains of bacteria, these bacteria that are really hard to treat because they're resistant to treatment with so many of our modern antibiotics, we usually expect to find them in hospital settings where really sick people are coming in to be treated, bringing their serious infections with them, and where ultimately selective pressures that we'll talk about here in a second are being applied that select for more resistant strains of bacteria. But that's not what happened with Addie. Addie encountered these really difficult to treat bacteria out in the community. So when we talk about community-associated infections, what we mean is that you encounter these bacteria, these difficult to treat bacteria, in places that the public commonly uses. So schools, playgrounds, grocery stores, the movie theater, places that are regularly used by members of the community in their day-to-day life. So she got this serious, difficult to treat infection just because it was present in her day-to-day environment. Now, things for Addie got pretty serious with this MRSA infection because she ended up experiencing sepsis. So in sepsis, the bacteria actually gets in your bloodstream. And that's a really big deal because it means the infection is no longer localized to where you initially experienced that infection, but instead now spreads all over your body because it's traveling through your blood. So at that point, Addie was in pretty big trouble. And the infection became so serious in her lungs in particular that she ended up needing to be put on an apparatus called ECMO, which is basically a machine that oxygenates your blood for you. Addie's lungs were so badly damaged that they couldn't adequately oxygenate her blood to carry oxygen to the rest of her body. So essentially what they had to do was put some big tubes in Addie's neck so that her blood could come out of her body when it was deoxygenated, run through the ECMO apparatus, which would oxygenate her blood, and then get pumped back into her body to keep her alive. Now, that's a really good thing that the ECMO could keep her alive. It's critical life support. But the problem with that ECMO apparatus is that you have all these tubes running through the machine and going into Addie's body. And it's really difficult to make sure that all those lengths of tubes are fully sterile. Excuse me. So a threat to using the ECMO apparatus is that a lot of times it introduces secondary infections and basically adds on another level of challenge for the patient. Now, there's nothing to do about it. If you need total life support, you have to go on the ECMO apparatus, but it does cause this additional risk of those secondary infections. And unfortunately, that's what happened for Addie. Addie ended up getting a secondary infection with a type of bacteria called Stenotrophomonas. Stenotrophomonas is a type of bacteria called gram-negative. And gram-negatives are really, really hard to treat. And we're going to talk about why gram-negatives are so much more challenging to treat on average than gram-positive bacteria. Now, ultimately, when we think about infection with Stenotrophomonas, the reason it's such a big deal is because, to begin with, under best case scenarios, Stenotrophomonas is only treatable with a couple different types of antibiotics. So normally when we think about approaching treatment to your standard bacterial infection, we've got a huge host of antibiotics that we can potentially apply, but that's not so for Stenotrophomonas. At the outset, it is resistant to treatment with most known antibiotics, but the doctors had to attempt to treat it with what they had available. So essentially what happened with Addie when she got this Stenotrophomonas is that her doctors started trying to treat her with each of the different antibiotics that is potentially usable against this serious gram-negative Stenotrophomonas infection. So what happened with Addie is that the doctors would apply an antibiotic to her infection and should start getting better. Her symptoms would recede and there'd be a lot of hope. But then the Stenotrophomonas symptoms would increase again and should go back to the totally debilitated, seriously critically ill state she was in before. The antibiotic stopped working. So they went through this process with each of the different antibiotics, treating her, she got better, and then the symptoms came back. Treating her, she got better, and then the symptoms came back. Eventually, they had tried every single antibiotic that can potentially be used to treat Stenotrophomonas infections until they were all out of antibiotics. And at that point, Addis stenotrophomonas infection was classified as pan-resistant. Pan-resistant means resistant to all known antibiotics. In other words, there was literally nothing left. Nothing in modern medical science was available anymore that could potentially treat this bacterial infection Addie had in her lungs. So at that point, the doctors really didn't have any options. The infection was in her lungs. They had no way to help her get better. Her lungs were so badly damaged that there was really no hope of those organs recovering. So the only option left was to transplant her. The only hope they had for treating Addie and helping her stay alive was to physically remove the infected tissue, both of her lungs, hope that they got all the bacteria out, and then give her new lungs and hope that she could survive the transplantation. So what we want to think about now, before we think about the end of Addie's story, is why all that happened. What are gram-negative bacteria, and why are they so incredibly difficult to treat? And that's what we're going to think about now. We're also, after we consider why gram-negatives are so difficult, is consider what was happening in Addie through the lens of evolution, considering both the origin of these serious resistance alleles and why Addie experienced the treatment progression that she did, where she'd get better with an antibiotic and then get worse, and get better and then get worse. So what we're going to do now is apply our knowledge of evolution to explain what was happening inside of Addie. So first, we want to think about the key differences between gram-negative and gram-positive bacteria, to think about why those gram-negatives, like stenotrophomonas, are on average so much more difficult to treat than gram-positive bacteria. When we think about the differences between these bacteria, the key differences are between cell wall thickness and then complexity in membrane organization. So what we're going to do is look at these two very simplified diagrams of the external anatomy of gram-positive bacteria on the top versus gram-negative bacteria on the bottom. So we're going to start with our gram-positives on the top. Gram-positive external organization is really simple. So if we start from the inside out, this green area is the cell membrane of that gram-positive bacteria. So every cell has a cell membrane, and this is just representing the cell membrane of these bacteria. On the outside of that cell membrane, you have what's called a periplasmic space. And that's just this little gap between the cell membrane and the cell wall on the outside. So this kind of bluish layer there is the periplasmic space. On the outside, this big, thick, kind of brownish layer, that represents the cell wall of our gram-positive bacteria. Peptidoglycan is the molecule that this cell wall is comprised of. And when we look at the thickness of this peptidoglycan cell wall in gram-positives, you can see it's really thick. That's a big, chunky cell wall. Now what we're going to do is compare it to gram-negatives like stenotrophomonas. Initially, the organization of those gram-negatives is really similar to the gram-positives. On the inside, you still have that kind of light greenish cell membrane separated from the cell wall by that periplasmic space. Now, when we get to that peptidoglycan cell wall, you'll notice a difference between our gram-positive and gram-negatives. Where the gram-positive cell wall was really thick, so lots and lots of peptidoglycan in that thick cell wall, the cell wall in our gram-negatives is much thinner. So we have a significantly smaller cell wall, less peptidoglycan in that cell wall. The other really key difference when we're starting to think about why gram-negatives are so hard to treat is that beyond that cell wall, we have an additional outer membrane. So outside of that cell wall, we've got another periplasmic space separating that cell wall from another outer membrane that encases that cell wall in gram-negative bacteria. That outer membrane is not present in gram-positives. So what we want to think about now is why we care about differences in cell wall thickness and that additional outer membrane that our gram-negative bacteria have. So the first thing that we're going to talk about is the relevance of that difference in cell wall thickness. And ultimately, we care about that because it's a really critical factor in thinking about how we identify a bacteria as gram-negative versus gram-positive. So when we talk about gram-negative and positive, the gram bit refers to the gram staining method. The Gram stain is a purple stain. And the way that that Gram staining method works is that that purple stain binds to that peptidoglycan in a bacteria's cell wall. The more peptidoglycan that's present, the more for that purple stain to cling to. So when bacteria are gram positive and have that really thick, chunky cell wall with lots and lots of peptidoglycan, that means there's lots of peptidoglycan for the stain to stick to. And gram positive bacteria show up this really deep purple when you stain them with this gram staining process. But when we think about that difference for gram-negative bacteria, we said that gram-negative bacteria have a much thinner cell wall, so less peptidoglycan for that purple stain to bind to. So when you wash gram-negative bacteria with that purple stain, there's not much for it to stick to. So they end up looking this kind of lightish pink color. So the nice thing about that difference between cell wall thickness is that it makes it really easy to identify whether a bacteria is gram positive or gram negative when we're trying to figure out the infection that a person is suffering from. Okay, so that's cell wall thickness differences. But the next thing that we want to think about is that outer membrane, and more generally, why it's so difficult to treat gram-negative bacteria on average with antibiotics. So the first thing that we have to consider if we're going to think about why it's hard to treat gram-negatives is what antibiotics actually do inside of you when you take them. So in general, when we think about antibiotics, the goal is to stop the bacteria from reproducing and living while not hurting you, the also living organism that's going to be exposed to these antibiotics. So in general, when we think about the action that antibiotics have, antibiotics act by binding with some specific surface receptor on the cell membrane of bacteria. If that antibiotic compound has the right shape to bind that targeted receptor, it can then have its targeted effect on that cell. So in general, when we think about what antibiotics are doing to bacterial cells, there are a couple different options. One is to have some effect that kills the bacteria outright, and two is the option that prevents that bacteria from reproducing further. Killing the bacteria outright can involve disruption of cell wall maintenance and cell wall building or cell membrane maintenance, or disruption of the bacterial ribosome, so preventing that bacteria from synthesizing proteins essential for living. Another option is that it can prevent the bacteria from reproducing itself so that that population can't grow, and after the current members of the population die, the infection is over. But either way, it stops the infection from proceeding by killing them or from stopping the bacteria from reproducing themselves. So what we want to do is think about why it's so much harder for bacteria to have that effect on gram-negative bacteria. Gram-negative bacteria have a bunch of different potential mechanisms of resistance, and we are going to do a not at all comprehensive list of them. Instead, we're just going to talk about the most common. The biggie that I want you to take away from this is that that outer membrane at the outset makes treating gram-negatives by default harder than treating gram-positives on average. That additional outer membrane that gram-negatives have, it turns out on average is highly impermeable to most of the antibiotics that we have. So when we talk about permeability of a membrane, we're describing how easy it is for stuff to move across that membrane. Based on those outer membrane characteristics, those antibiotic compounds have a really, really hard time making it from the outside of that outer membrane and penetrating into that outer membrane so that they can get to those surface membrane receptors that they have to bind to. So it's basically like a coat of armor surrounding that bacteria that keeps the antibiotic compounds out. If the antibiotic compounds can't make it in past that outer membrane to bind to those receptors, then they can't have their effect on the bacteria, and the bacteria is not treated. So this is a major mechanism of resistance for gram-negative bacteria. But unfortunately, for a lot of gram-negative infections, that's not even all there is. We also know that they experience regular surface receptor mutations. So essentially, if we think about binding a receptor, if we imagine my hand is the receptor and my fist is the antibiotic compound, I'm going to make sure that you can actually see what I'm doing here. I had my video turned off, so I wasn't distracted looking at myself while we were recording. So if we imagine my hand is the receptor on that cell surface and my fist is the compound, it has to fit with essentially a lock and key mechanism. But what we're saying is that that surface receptor can change its conformation. So maybe it gets a lot smaller, and all of a sudden that antibiotic compound can no longer bind with that receptor that has a different shape and therefore can no longer treat that infection. And then last but not least, again, in a by no means comprehensive list, is that sometimes what we see is the development of antibiotic efflux pumps. So if that antibiotic compound can make it through the outer membrane, bind with whatever that surface receptor is, some bacteria have other surface proteins that actively pump that antibiotic back out before it can have its effect. So lots of really difficult features of these gram-negative bacteria. So what we want to do now is examine Addie's gram-negative Stenotrophomonas infection through the lens of evolution to think about why her infection proceeded as it did. So what we're going to do is think about this in the context of those population phenotype frequency diagrams that we've already worked with. So what we're going to do is examine the progression of the bacterial population of Stenotrophomonas inside Addie. So when we think about the initial bacterial population, we can imagine the distribution of a given phenotype for resistance in her initial bacterial population. So I picked permeability to antibiotics. On one end of the axis, we have those individuals that would have a really high permeability to antibiotics. So relatively easy for that antibiotic compound to move across that outer membrane and bind with the relevant surface receptors. On the opposite end of the spectrum, we have those individuals with really low permeability to antibiotics. So those where it's really, really difficult, if not impossible, for that antibiotic compound to move through that outer membrane and bind the surface receptors. So essentially, we have a continuum of really easy to treat bacteria over here and really difficult to treat bacteria over here in Addie's Stenotrophomonas population. So what this figure depicts is the number of bacteria exhibiting these different permeability characteristics before she was treated with an antibiotic. So at this point, Addie is really, really sick. She's got this big giant bacterial infection that's causing disease symptomology and is damaging her tissues. So at that point, what our doctors do is apply a selective pressure to the bacterial population, which is an antibiotic. So when the doctors start giving her those antibiotics, the most susceptible to that environmental pressure die quickly. So we start killing off portions of her population. And as we continue to apply that selective pressure on the bacterial population, more and more of them die as we build up that effective dose of antibiotic. So eventually, when we're finishing out that course of antibiotics, most of the bacterial population is gone. So at this point, we have a very small number of individuals left who are highly resistant to treatment with antibiotics. So let's think about what this means for the next generation of Addie's infection. At this point for Addie, she's feeling pretty darn good. There are very few individuals left in her population. The survivor population is quite small, and there are few individuals causing disease symptoms. So Addie's feeling really good. It looks like she's been cured of her infection. But what we want to do now is apply our knowledge of natural selection to think about what this means for the next generation. If these are the survivors of that selective pressure, and assuming this trait is heritable, which it is, that is a realistic assumption for these resistance phenotypes, what does the next generation look like when these surviving individuals reproduce? Well, unfortunately, if all of the survivors have alleles that give them high resistance phenotypes, then the next generation has a shift in their average resistance phenotype, where all of the individuals now exhibit some degree of high resistance phenotype and are all extremely difficult to treat with antibiotics and are impossible to treat with that antibiotic pressure that's already been applied. So at this point, when those survivors have reproduced and that population swells back up to its pre-treatment levels, Addie gets sick all over again. So at this point, the doctors then attempt to apply another selective pressure in the form of a different antibiotic. And Addie goes through that process of selection on that population of bacteria living inside her again and again, where they kill off all but the most resistant. Then those survivors reproduce until eventually she is left with a population of Stenotrophomonas living inside her lungs that are resistant to every single known antibiotic. So when we take a look at what happened in Addie, what we're doing is watching selection play out in that bacterial population again and again and again, until essentially the doctors ran out of selective pressures to apply to those bacteria. And at that point, the only thing they had left to try with Addie was physically removing the lungs through transplantation. Before we move on to thinking about the next example of antibiotic resistance playing out in another individual, what we want to do is also think about how these antibiotic resistance alleles developed in the first place. So what I want you to do now is apply your understanding of mutations. So go back to that definition of mutation, critically assess the key components of mutation, and then read over these options to pick the one that correctly describes how that mutation in the bacteria that if infected Addie initially occurred. So how did we get antibiotic resistance alleles in the first place? So go ahead and pause the video, head on over to Top Hat, enter in the correct answer, and when you're done, come on back over for a little debrief explanation. Okay, so hopefully what you figured out is that the correct answer is that the mutations that resulted in resistance to each antibiotic likely existed in the population before Addie received the antibiotics. So let's think about why that's the only reasonable option. When we think about mutations, a key portion of that definition of mutations is that they are random permanent changes in the DNA sequences of an organism. So when we think about options A and C, both of those options are suggesting that the bacteria developed a beneficial mutation because it would be helpful for their survival and reproduction in response to an environmental pressure. But we know that's never the case. Because mutations are random, individuals will never develop a beneficial mutation in response to a selective pressure. So when we think about the mutations for resistance that were present in Addie's Stenotrophomonas population, those resistance alleles were very likely already present in the population. Now, prior to treatment with antibiotics, those resistance alleles were essentially neutral variation. Absent the pressure of an antibiotic, having resistance alleles was neither beneficial nor detrimental. But when you apply those antibiotic pressures, all of a sudden having those resistance alleles becomes super beneficial, and they increase in frequency as natural selection proceeds in that population. But critically, those mutation alleles happened randomly. Now, of course, we can't say with 100% certainty that they existed prior to antibiotic treatment because they're random, right? We don't know when they happened. They might have randomly popped up after treatment with antibiotic resistance. But if we think about the probability of that, it's extremely unlikely. The much more likely scenario is that those random mutations existed in the population prior to any treatment with antibiotics. Okay. So the next portion of the video follows a different case study about a young man named David. And we're going to talk a little bit about what happened with David before getting to the critical portions of his story, considering another aspect of how antibiotic resistance is spreading and the nature of this spread. So the second portion of the documentary follows a guy named David Ricci, and ultimately David Ricci was visiting India, and he and his group were walking from one portion of the city to another when David got hit by a train. So he ended up getting pulled under the train, and the train ran over his leg, and they don't show that in the video, obviously. But David was in really serious condition and had to be rushed to a hospital where he had to have the remains of his leg amputated. And that's obviously really bad, but the big difficulty for David came in when it turned out that that site of amputation had become infected with bacteria carrying a gene called NDM1. Now, NDM1 is not a strain of bacteria. NDM1 is an allele that carries significant resistance information. Now, the really tough part with NDM1 that our video describes is that NDM1 can be spread from bacterium to bacterium. And not only can it be spread from one bacteria to another within the same species, it can also be spread from species to species. So bacteria can communicate this resistance information to one another. So for David, when he had that initial infection in the wound site of his leg, he was infected with lots of different species of bacteria. Some of them started out easily treatable. But since some of the bacteria in his wound carried this NDM1 allele, what his doctors found is that those previously treatable strains of bacteria rapidly became untreatable as they gained this NDM1 resistance allele. So kind of like what happened with Addie, at that point, David had all of these untreatable infections in his leg that were pan-resistant, completely resistant to treatment. Now, for David, one of the things that the doctor tried was basically rewinding the clock and going back to some of the earliest developed antibiotics that we hadn't used for decades. And the reason we hadn't used them is because, one, well, this was a good thing. They were really powerful in killing bacteria, but they also had serious negative side effects on humans. So we had moved away from them because their side effects were so damaging to a lot of the people we were using them on that you ran the risk of the antibiotic killing the human in the process of killing the bacteria that we were trying to treat. And ultimately, that's what happened with David. The toxic side effects of that bacteria, colistin, that they were trying to use on him became so severe they had to pull him off it because he was going to die if they continued attempting that treatment. So just like with Addie, they had run out of options to treat David's antibiotics. And just like with Addie, the only option they had left was to remove more and more of his leg in hopes of physically removing the infection with the tissues that contained those bacteria. So what we want to do now, before we think about how we got to a scenario in modern medicine where our only option was cutting infected tissue out, is we want to think about the NDM1 resistance information and this difference in how that resistance information was being spread among bacteria. So typically when we think about resistance information and genetic information spreading among populations, we think about it in the way that we just described with Addis stenotrophomonas. So parents pass genetic information onto their offspring. Like Addis stenotrophomonas, the survivors with resistance allele passed that resistance information onto their offspring. But that's not what happened in David with NDM1. When David's bacterial populations of different species interacted with one another, those pre-existing individuals transferred genetic information to one another. So this is a really different thing, and we call it horizontal gene transfer. In horizontal gene transfer, you have the movement of genetic information among organisms without reproduction. So two pre-existing individuals can exchange genetic information with one another such that the genetic information contained by one individual changes, and they can express novel phenotypes within their lifetime without any reproduction. So let's think a little bit about how that works. When we look at a really simplified depiction of the genetic information contained inside of a bacterium, they have ultimately two different types of DNA. The first is the main chromosomal DNA. So that's what we've got here. This has all of the information critical for survival and reproduction. So stuff that they absolutely have to survive and reproduce. The other thing that bacteria have, oops, I didn't mean to move forward. The other thing that bacteria have is plasmid DNA. Plasmids are additional pieces of DNA that are separate from the main chromosomal DNA. So just like our chromosomal DNA in bacteria is not linear, like it is in us, it's circular, so too are the plasmid DNA molecules. So we have these small circular chunks of DNA that are separate from the main chromosomal DNA of the bacterium. That plasmid DNA is typically where we find information about resistance in bacteria that are resistant to antibiotics. So what we're going to do now is consider how this NDM1 allele that confers massive resistance information can spread from bacterium to bacterium. Okay, so what we're going to do is start out with two different bacteria individuals here, a donor and a recipient. So what we're going to do is imagine that this donor has that NDM1 allele on this plasmid that we can see in the picture. So ultimately, we have a donor that has this resistance information and a recipient who doesn't. If that donor bacterium comes into close proximity to another individual, it can ultimately develop what's called a sex pilus. So basically this bridge from one bacterium to another. So that donor forms essentially a bridge to another individual. Once that connection between donor and recipient is made, one strand of that double-stranded plasmid DNA unwinds and moves from the donor individual into the recipient individual. So at that point, the original donor now has one strand of that plasmid DNA with the resistance information, and so too does the recipient. So once they each have a copy of that plasmid information, they go through the typical process of DNA, complementary DNA production, where they just use that sequence of nucleotides to produce the complementary strand. And at that point, both the original donor and the previous recipient now have double-stranded plasmid DNA that have that resistance information. So what that means, if you started out with only one individual capable of transferring that resistance information, you now have two fully resistant individuals, both of whom are capable of passing on that resistance information to other bacteria that they encounter. So what happened in David is that you had some individuals with that NDM1 allele that conferred massive resistance information, and they passed that resistance information on to all the other individuals in his bacterial populations, both within the same species of bacteria and between species. So his previously treatable bacterial populations became fully resistant to all antibiotic treatments because of this sharing of resistance information through horizontal gene transfer. Now, this is really bad news for David, obviously, because it means that we can't treat his infections with anything other than amputation. But from a public health crisis, NDM1 sharing turned out to be quite a bit more serious. Typically, when we think about the sharing of resistance information through horizontal gene transfer, it's localized to an individual patient. So usually we see an individual who might be infected with a highly resistant strain of bacteria, including bacteria carrying that NDM1 gene, passing that resistance information to other bacteria in the patient. Now, for that individual, obviously that's really serious because it means it's much harder to treat them. But from a public health perspective, that's less of a threat because it's localized within an individual and therefore won't affect other individuals who aren't exposed to that infection. But for NDM1, what doctors in India found is that that resistance information was present and being shared out in community spaces where sanitation was poor. So when they went out and looked at puddles in areas where sanitation was poor, they found bacteria just in regular puddles on sidewalks and streets around people's home. They found bacteria that had that NDM1 allele, and they found that sharing of NDM1 was happening in public spaces. So that's much more serious because it means that that resistance information is spreading in bacterial populations in a much broader context than we previously realized was happening. It also means that a much broader segment of population is likely to encounter those highly resistant forms of bacteria. So now that we've thought about what was going on in Addie and in David, we want to consider how we got here in the first place. It's pretty shocking to consider that in the 21st century, we can experience a situation where sophisticated modern medical science can't treat bacterial infections. And we get to the point where the only option is literally to cut the infection out of people. That seems really primitive almost relative to modern medical technology. It's something you would have expected us to be doing 100 years ago, but certainly not in the 21st century where we're so sophisticated in our medical technology. So what we want to think about is how we got here. If we rewind the clock less than 100 years ago, we had no antibiotics. But through scientific innovation and technological advancements, we went from a world with no antibiotics to the world that you and I grew up with, where it was more or less unthinkable that you could die from a bacterial infection. So the question that we want to address now is why? Why did this happen? Why aren't we just continuing to innovate and develop antibiotics to treat this next realm of really difficult to treat bacteria, where that is a pressing modern medical need? Why aren't we innovating more to solve this problem? So that's what we're going to look at next. And so for this one, I'm going to briefly stop share that so that I can optimize for video. We are actually going to go back to that documentary because this portion is mostly talking about the pharmaceutical side of things. So what's going on with pharmaceutical companies and pharmaceutical company decision making that's resulting in us experiencing this crisis. They are going to talk about medical situations here, but the imagery is not challenging from a medical standpoint. It's mostly discussing the pharmaceutical companies and their development of antibiotics and why that development hasn't proceeded in the way we're used to. At the very end of the documentary, they're going to revisit David and Addie and talk about what happened with them at the end of their course of treatment. They will be showing portions of David's amputated leg, but the healed leg, so a fully healed leg, not anything from his medical situation. So we're going to go ahead and get started with that. It should be optimized. If something goes weird with screen sharing, you're just going to pick up with the documentary at minute 33. So here we go.
The prospect of life without antibiotics is barely.
Oh, hopefully it starts.
Full supply of them since the end of World War II. They are a staple of modern medicine. It's hard to recall a time without them, when an infected cut could kill a healthy young person in a matter of days. But it's now clear that we are heading back in that direction, that the miracle of these drugs is slipping away.
Antibiotics are unique drugs. They're not like any other class of drugs. 50 years from today, the cholesterol drugs we have now will work just as well as they work today. The cancer drugs we have now will work just as well as they do today. That's true of all the other drug classes. Antibiotics are the only class of drugs that the more we use, the more rapidly we lose. When you use it, it becomes less effective for me and vice versa.
That is the essence of antibiotic resistance.
The more you expose a bacteria to an antibiotic, the greater the likelihood that the resistance to that antibiotic is going to develop. So the more antibiotics we put into people, we put into the environment, the more opportunities we create for these bacteria to become resistant.
But people forgot about the danger of resistance because the drugs were so effective.
And what they had forgotten was the warning that Alexander Fleming himself, the man who discovered penicillin, gave us in 1945, that resistance was already being seen, and the more we wasted penicillin, the more people were going to die of penicillin-resistant infections.
Bacterial resistance is largely inevitable, but it's also something that we have certainly helped along the way. We have fueled this fire of bacterial resistance. These drugs are miracle drugs, these antibiotics that we have, but we haven't taken good care of them.
Public health officials estimate that 1/3 of all antibiotic use in the U.S. is either unnecessary or inappropriate.
And in overusing these antibiotics, we have set ourselves up for the scenario that we find ourselves in now, where we're running out of antibiotics.
But the growing scarcity of effective antibiotics isn't just a problem of overuse. It's also been driven by what's happening inside the drug industry itself.
The place where it started to turn really challenging, I'd say, would be in the 80s and the 90s, when we began to see occasional bacteria that were very hard to treat. And it became less obvious that you were able to invent new antibiotics. And the brand new things just weren't coming at the same pace. And then in the 90s and the first part of this century, we began to see resistant bacteria for which we really didn't have very much or anything at all, and we had nothing coming to treat them.
That's because most major drug companies were pulling out of the antibiotic research field just as the gram-negative threat was worsening. One of the last companies to stay was Pfizer, which had made its name on antibiotics. By the mid-2000s, it had set its sights squarely on the gram-negative problem.
We thought there was medical need. That's really matters. And we thought that given our history in being able to develop penicillin, the antifungals, antibiotics, that in fact, if we put our minds to it, that we would succeed. But this is a highly risky and unpredictable enterprise.
Despite the risk, Pfizer built a world-class research team in Groton, Connecticut, and brought in a veteran in gram-negative research, John Quinn.
In 1983, when I finished my training, almost every pharmaceutical company had an antibiotic development team. And by the time I landed at Pfizer in 2008, we were really down to 3 big guys and some smaller companies, biotechs and so on. And I think all of us felt that, we had a moral obligation to continue to work in this area. There was a pressing clinical need. Most companies had abandoned the field, and we were still in the game. We were proud to still be in the game.
Quinn and his team believed they were onto something big, several different compounds to treat gram-negatives. The potential breakthroughs got the attention of the company's science advisors, including Brad Spellberg.
I felt that their pipeline was probably the most comprehensive and important antibacterial pipeline in the world, focusing on the types of bacteria that we're really having severe problems with right now.
Which are
the highly resistant gram-negative bacteria. These would have solved problems and saved lives had they been successfully developed.
But bringing these drugs to market faced the economic paradox of antibiotics.
If you need an antibiotic, you need it only briefly. Indeed, that's the correct way to use an antibiotic. You use it only briefly. And from an economic standpoint of a developer, that means you're not getting the return on the investment you've made, because you've spent between $600 million and $1 billion to bring that new antibiotic to market.
Wait, it costs up to $1 billion to bring a new drug to market?
It can easily cost up to $1 billion to bring a new drug to the market. And the initial reaction to it is, that's great, and let's not use it. Let's use it as little as possible.
So here's a large company saying, I can make billions off cholesterol drugs, blood pressure drugs, arthritis drugs, dementia, things that I know patients are going to have to take every day for the rest of their lives. Why would I put my R&D dollars into the antibiotic division that isn't going to make me any money when I can put it over here?
So here's the deal.
That's going to make a lot of money for the company. I answer to the shareholders.
That was the problem facing Pfizer in 2011.
Don't kid yourself. Talk to your doctor.
Its stock had plummeted on Wall Street, and its blockbuster cholesterol drug, Lipitor, was about to lose its patent.
I received an email on my BlackBerry that there was a mandatory emergency meeting in 2 hours. Can't be good. So I called in for the meeting and was told that the announcement had been made that the Groton facility was going to be closed.
The company ended 70 years of leadership in antibiotic development, leaving its search for a gram-negative cure unfinished.
The external people who I spoke to, many of whom are my personal friends, said to me, well, Pfizer's just doing what other companies have done. There's nothing particularly wrong with that. It's not immoral. We are a capitalist society.
In 2013, we asked Pfizer to explain the decision.
I get the sense that you have to make some very ruthless decisions about where to put the company's capital, about where to invest, where to put your emphasis. And when you pulled out of gram-negative research like that and shifted to vaccines, do you look back on that and say, you know, we learned something about this?
These are not ruthless decisions. These are, portfolio decisions about how we can serve medical need in the best way. We want to stay, in the business of providing new therapeutics for the future. Our investors require that of us. I think society wants a Pfizer to be doing what we do in 20 years. We make portfolio management decisions.
In 2016, Pfizer decided to re-enter the antibiotic market and bought several drugs under development. Like other large pharmaceuticals, though, it is still not investing in research.
There is an increasing recognition that antibiotics are not a good thing to run off a pure capitalistic market, okay? We need to switch from an entrepreneurial business model where you maximize sales to other payer mechanisms, where the goal is society can say, we want these kinds of antibiotics developed, and we're going to help you out. We're going to decrease your cost and risk. In return, on the back end, we're going to have some say in how it's used so it doesn't get abused.
In Washington, the federal government has been ramping up its involvement in the superbug fight. 2 years ago, the Obama administration unveiled a national plan coordinated by the Department of Health and Human Services. The point person for the effort now is Christopher Jones.
Under the National Action Plan for Combating Antibiotic-Resistant Bacteria, which was released in 2015, we now have an overarching framework and structure for addressing this issue across the federal government.
We have a plan.
We have a plan that has specific actions that are being worked on every single day, and I think it's reflective of the investments we've been making around surveillance, stewardship, developing new products, developing new diagnostics, and increasing international collaboration on the issue.
For a long time, people have been warning that the pipeline of new antibiotics is running dry. How's that going?
We're investing $250 million over 5 years for the early stage development of antimicrobials. I think the next phase, as we start to think about products that really show promise in early phases, is how do we continue to support their development? And I don't think we've fully landed on what that strategy looks like, but we do have to rethink how we reward companies. And that's, again, ongoing conversations.
But some say the government should be playing a larger role.
Is there more that needs to be done?
Where we need to focus on now is using less antibiotics. We need to create policies and regulations. If we publicly reported antibiotic use, attach requirements to hospitals, you have to report, that public shaming effect will drive antibiotic use down.
And these are all the M28s?
Yes, sir.
Even as the government's strategy is taking shape, the Trump administration has proposed funding cuts of up to 20% to programs and agencies that combat antibiotic resistance.
What will be the significance and the impact of new budget reductions in antibiotic resistance?
To your point, yes, a lot of what we're doing requires investments. It requires resources. If there is less money to spend, there are tough choices that have to be made, things that can't be done.
I'm very concerned about it, and I think we all should be. I think the gains that we have made have been largely because of investment. And if we cut back, you're going to see an even faster evolution of resistance and spread of resistance, and way fewer countermeasures being developed to combat it.
Last year, the warnings became even more dire.
Drug-resistant bacterial infections are on track to kill more people than cancer.
One international report predicted
by 2050, superbugs could kill 10 million people a year.
Now we are seeing bacteria that are resistant to the absolute last stop on the train, colistin. And so for those patients, there are no options left. So those patients are truly have gone back in time. They are back in the pre-antibiotic era, and they will recover from those infections or will die from those infections, and there's nothing we can do for them.
As we reported in 2013, NIH never did fully rid itself of the deadly superbug KP.
So for this very small portion, it's going to talk about KPC for about 1 minute. We skipped over that in the earlier portion of the documentary, so don't worry about the KPC bit at NIH.
A year after the outbreak, a young man came to the hospital because of complications from a bone marrow transplant. While he was there, he contracted KPC and died, the 7th victim of the outbreak.
I guess if I had a major message, it would be that it's never going to end. So this organism and organisms like this are going to be with us till the cows come home. And we have to learn how to deal with them. We have to change our culture in the hospital.
KPC has been found in hospitals in all but 2 states, and that's just the hospitals that are voluntarily reporting it.
This is the portion where David will eventually be showing the healed portion of his leg at the amputation wound. It is fully healed, but if that's challenging for you, I wanted to let you know.
As for David Ricci, it took 3 surgeries and another round of highly toxic antibiotics before doctors believed they had removed all the NDM1 from his leg.
You know, there's no muscle left on it, and I only got about 6 inches left, and the bone stops there. And
so far, Ricci has remained healthy, though not entirely free from the fear of NDM1.
You know, my doctors were pretty straightforward with me. They were very honest and said, you know, there is a good chance that this infection might not go away.
Might not ever go away.
Yeah. They said, you know, we don't have enough experience to know what's going to happen.
NDM1 has now spread to at least 70 countries. And here in the U.S., more than 200 cases have been reported.
So David was actually sort of a harbinger of something to come.
David was an example of something that's already here. So there are entire continents that have this major problem, a public health problem already. David was simply a sample of that population, new to us. And that's key because hospitals in any city in the country are going to have patients from all over the world. That globalization, that mobility is going on now. This is already here.
All right, Addie, let there be light. This is the day that the Lord has made.
Addie Rarasich was finally able to return home.
Yeah, but it's like not fun. Let there be no light whatsoever.
She received the double lung transplant she'd been waiting for.
It was like bringing home a premature baby. We brought home monitors, and she couldn't do anything for herself. She couldn't even turn over in the bed. She couldn't turn side to side. That's how weak and contracted and debilitated she was.
So how are you doing now?
Basically, I'm fine. Nothing seems out of whack right now. I feel pretty good. I look pretty much like I did before. I have all my friends back.
Did you understand what was happening to you?
No.
Did anybody talk about infection and what infection meant?
No. Basically, what I was told is I'd say I want to go home. And she'd say I couldn't make the drive home ever. Like, I was too sick to go home.
Okay. Everything's hard for Addie now. Everything's a battle.
Prescriptions.
She has to take a handful of pills twice a day. We have to worry constantly about, picking up a bacteria or a virus. She's had pneumonia 5 times. Bacterial pneumonia. that had to be treated with antibiotics. And every time I wonder, is this the time that we're going to come up against a bacteria that they don't have anything to treat it with? I think for lung transplants, the survival rate, about 80% make it a year, and about 50% make it 5 years. And every year after that, the risks just go up.
Let's bow our heads in prayer.
But people might say, the story of Addie is horrible, but that won't happen to my daughter. Could this happen to anybody?
It happened to Addie. She was healthy. It could happen to anybody. It could happen to your next door neighbor. It could happen to your child. It could happen to anybody. Now, I'm not here to practice doomsday thinking, but those bacteria are out there. And they're out there in healthy people in the community.
If you don't mind standing up, you can walk around. Can we? Please greet each other and pray.
The average person thinks, oh, I have an infection, I take an antibiotic, I get better. Yeah, it's not that simple anymore.
Morning, Caleb.
How you doing, sweetheart?
I'm good, thank you.
Addie didn't get better, did she?
No, she never did get better. Really. She didn't. She had to have surgery and take the infection out, but...
May have saved her life.
For now. Bought her time. That's what happened. We bought her some time, and I am grateful for every minute of it.
Nice to meet you. Hi, I'm Maddie. I don't think we ever met.
I remember you. I know I've seen you somewhere before.
Yeah, me too. Were you my nurse?
Yeah.
All right. So, excuse me. What we're going to do now is think a little bit more about what this means for where we are in terms of our thoughts about antibiotic availability, antibiotic development, and the impacts that it has on us as a country, and then ultimately us globally. So like we saw in the video, the issue with the development of antibiotics for gram-negative bacteria isn't that the science or technology can't keep pace, but that, excuse me, but instead is more of, let me make sure that my Zoom is still sharing screen. Okay, yes, I am. It looks funny when you share for optimizing video, so I wanted to make sure. But is more of an issue of pharmaceutical companies deciding to pull out of research on developing antibiotics for gram-negative bacteria. So what I want you all to think about, not only is a lot of you future members of the medical community, but also just as people who will have loved ones or yourself who might get sick with an antibiotic resistant infection, hopefully not, but something that all of us really need to be thinking about. So I want you to do is consider whose responsibility it is to make sure that these health crises like antibiotic resistance are addressed. And the way that we're going to do this is with a couple different things. So the first thing that I want you to do is just on whatever word processor you want on your computer, iPad, whatever, is write up answers to these questions. Was it ethical for the pharmaceutical companies to pull out of gram-negative antibiotic research? And whose responsibility is it to ensure that health crises like these are addressed? Now, the critical bit of these questions isn't your thoughts on it, like yes, no, but instead your reasoning for your answer. You will get credit for whatever you say in terms of whether you think yes, no, it was or wasn't ethical and who's responsible, as long as you do that second bit of each prompt. Justify your answer. I'm not asking these questions to get you to parrot my thoughts. And you'll notice I've not told you my thoughts. What I want you to do is think about the information that you've heard, think about the information that you know, and then consider what that means for your thoughts on these questions. Then you should be able to justify why you think what you think. So what I want you to do is go ahead and write that up. And as soon as you do, you're going to go to our Carmen page and go to assignments and locate the assignment titled ICA Antibiotic Ethics. And you are going to submit your assignment there. This is an individual assignment, so please do make sure that each person submits it. If you watch this with somebody else, that's totally fine. Just make sure that you both submit something and you can talk about it, no problem. The other thing that we're going to be following up with in class, just to stimulate a good discussion, is a top hat question where it's just a simple yes, no to this first option about pharmaceutical decision making. I know that if you're watching this on your own, you probably won't have that chance to have that conversation, but I don't want you to miss out on those top hat points. So go into top hat, answer that question about whether or not it was ethical, yes or no. Again, because this is an opinion one, there is no right or wrong answer. They're all coded as correct. So I just want you to put in the one that best reflects your general feelings. I know it's a complicated topic, and yes, no certainly can't summarize it, but it's just meant to be a starting point for discussion. So after you do that, you are done with this unit investigating antibiotic resistance through the lens of evolution, and you'll be good to go. So thanks, everybody. Have a really good day, and I'll see you all later.